Okay, go ahead. Thank you.
So the motivation for the plant cell atlas is really understanding the location to function paradigm. So it's similar to the idea of like structure to function. If we know the structure of something, we can sort of make predictions about what its function is. The idea is if we know the locations of biomolecules, we can predict what their functions might be. And single cell approaches are really rapidly expanding our knowledge of where these biomolecules are in plants from the molecular level all the way up to the organ and organism level, which is discussed in this white paper written by the Plant Cell Atlas group. And so we can make these observations with these single cell technologies, but the real question is then, how do we test these predictions about function that we come up with based on location? With this question, there's a lot of limitations to traditional genetic approaches. So if you make a full knockout mutant or you overexpress constitutively a gene, you may not be able to support the importance of that gene's location to its function. So if you do have a gene that's indeed very specifically expressed in one place and you knock it out, then maybe you are studying what it's doing in that place. But sometimes we have genes that have multiple roles and are located in multiple places, and we really want to understand location-specific functions. You might be able to do that. You might say, oh, I have a marker. I have a promoter that I can express this in a specific cell type. But this does not give you a lot of control. So this might be sufficient to put the gene there, but the abundance might not be a relevant level for your study. So overall, we really can have a lot of pleiotropy or just be ineffective to study genes that have very context or location specific activity. And this is often the case for developmental regulators or other like environmental response genes, where we really do care a lot about these types of genes. And so having other tools in our toolkit is really important. And so synthetic biology tools can help us test location to function hypotheses. And I'm just highlighting this example here from Trevor Nolan, where they used single cell data to understand how plants respond to brassinosteroid. And so he found an interesting response really specific to the cortex cells, but wanted to manipulate specifically those cells. And so he used a cell type specific CRISPR approach to knock out a brassinosteroid signaling molecule, which is really constitutively expressed, but he knocked it out very specifically in the cortex to show that the cortex response was indeed important for the phenotype. Another thing is that the knowledge of location can help us engineer plants for specific goals. And so this is an example from Jen, where she took this SLR mutant. So SLR is a dominant mutation that causes shorter root length, impaired root hair development, and no lateral roots. But by by expressing this dominant mutation in specific cells with this promoter, which marks the lateral root primordia, which I think Sarah talked about using the same promoter yesterday, or the root length is no longer affected, the root hairs are no longer affected, but you maintain that loss of lateral roots. And then by tuning the amount of this protein with a gate method, a synthetic gate, she's able to get a quantitative variation in lateral root density. And so this is really important because some of the biggest goals that we have in plant biology will require precise spatial control. And so I've highlighted 3 here that have really attracted a large amount of funding, but a lot of goals that plant biologists have will require this level of control that we need these synthetic tools to create. So one example is from the Salk Institute. They're trying to develop plants that have deeper roots with more suberin to sequester carbon. The ENSA project, mostly, which is international collaboration, but a lot at Cambridge University, is trying to engineer plants that don't form nodules, which are these symbiotic organs, to form these symbiosis. And this requires a really carefully orchestrated developmental program to cause the nodule to develop and the symbiosis to initiate. And then another example is the C4 rice project, where scientists are trying to get rice to partition its photosynthetics apparatus into the mesophyll and bundle sheath cells to be a C4 plant and improve its photosynthetic efficiency. So with this background, I'm just going to finish with sort of our goals for the hackathon. First, and this is sort of, I think, helpful to think about when you're thinking about your project, what sort of what we're looking for. So the very first thing is that we're hoping to expand the expertise of plant single cell biology researchers, which is, in theory, you all, I know you have a variety of different expertises, to include new methods to perturb gene activities in specific cellular contexts. We'd also like to provide resources. So this is plasmids, plant transformation, to demonstrate the manipulation of gene activity in specific cell types. So can we use some of these tools that a lot of the mentors here have developed to manipulate something that you're interested in? And then finally, there are certainly some technological gaps for plant synthetic biology. And so if we're talking about building a circuit and for your problem, and you realize, well, exactly what I need doesn't exist right now, it would be really cool to come up with a new idea to demonstrate a new type of circuit or a new tool. And I think that's it for me.